Welcome to Translational Oncology Insights. I'm Rittika, and today we are examining one of the most challenging frontiers in cancer immunotherapy, extending CAR T cell efficacy to solid tumors. Joining me is Dr. Sneha Pathak, a cancer researcher preparing for postdoctoral work in California and the broader United States. Dr. Pathak, CAR T cells have achieved remarkable success in hematological malignancies. Yet, solid tumors remain largely refractory. What fundamental differences in tumor biology create this sort of therapeutic division in terms of tumor?
Thank you, Rittika. Hope you're doing fine. So the disparity for this stems from architectural and microenvironmental complexity that is absent in liquid tumors. On the other hand, the hematological malignancies present isolated tumor cells in circulation, thus allowing the direct contact of CAR T cell engagement. Whereas in solid tumors, they create a fortress-like structures with physical barriers, dense extracellular matrices, aberrant vasculature, and also elevated interstitial pressure that actually impedes the CAR T cell infiltration. More or less, the tumor microenvironment actively suppresses immune function through multiple redundant mechanisms known so far, and which has evolved to maintain tissue homeostasis, but can be hijacked in the malignant state.
Yeah. So let's dissect these barriers systematically, okay? So my first question would be, how do physical impediments specifically limit CAR T cell function?
Okay. So if we're talking about physical impediments, so in solid tumor, the architecture creates what we term as immunological deserts. Now, hyaluronic acid accumulation and collagen cross-linking generate a dense stromal network that physically exclude T cell infiltration. Also, simultaneously, aberrant angiogenesis produces tortuous like leaky vessels with very poor perfusion, thus also limiting the CAR T cell trafficking to the tumor sites. But physical barriers are compounded by metabolic constraints also. Rapid tumor cell proliferation actually creates a glucose depleted, lactate rich hypoxic microenvironments, and CAR T cells engineered for high metabolic activity, they become functionally exhausted in such conditions. The metabolic competition isn't just energetic, it's immunologically suppressive as well. Okay, and also the lactate accumulation directly inhibits T cell effector functions through its pH mediated mechanisms and competitive inhibition of glucose transporters. So all in all, the physical barriers aren't just due to the vasculature or the dense your ECM. It is also because of the metabolic, very strategic metabolism that is going on in the solid tumors, which actually inhibits the effectiveness of CAR T cells to
that was a very well-rounded answer. I furthermore wanted to ask, how are researchers addressing these kind of metabolic challenges that keep coming up in this domain?
Okay, so after the discovery of this phenomena, there are several elegant approaches emerging. So metabolic reprogramming through genetically engineering is something that is sought after in today's research fraternity, wherein genetically engineered CAR T cells are made so that they are capable to utilize alternative fuel sources like fatty acids for oxidation, for instance, which is less dependent on glucose availability because glucose is totally exhausted by the tumor cells, just as we saw a few moments before. Also, the other strategies involve co-expressing metabolic enzymes like catalase to neutralize ROS or the reactive oxygen species produced, or engineering the enhanced mitochondrial biogenesis to improve energy capacity in hostile environments is also being researched right now. That's it. Yeah.
Beyond physical and metabolic barriers, let's say, how do cellular components of tumor microenvironment actively suppress such CAR T functions?
Okay. Okay, so this is a very good question. So the immunosuppressive cellular network is remarkably sophisticated in such solid tumors. So tumor-associated macrophages polarize toward M2 phenotypes that secrete interleukins like interleukin 10, TGF-beta, CCL22, CCL17, chemokine gradients, thus establishing a sustained immune tolerance. Also, myeloid-derived suppressor cells deplete arginine and cysteine, amino acids which are essential for T cell activation and proliferation. But the most insidious component in cancer-associated fibroblasts, these cells create physical barriers. So the fibroblasts, which are present in the solid tumors, they kind of create a physical barrier through their collagen deposition while secreting CXCL12, which sequesters CRCR4, CXCR4 positive CAR T cells in stromal compartments away from tumor cells. They also express FAP, that is fibroblast activation protein, which has become an attractive target for dual CAR-T approaches today.
Right. Would you want to elaborate on this said dual targeting strategies, please?
Yeah, we can elaborate. So the dual CARs actually represent a sophisticated approach to microenvironment remodeling. So now FAP targeted CARs can eliminate immunosuppressive fibroblasts, while tumor antigen CARs can target the malignant cells. So, sequential or simultaneous administration creates one-two punch. First, dismantling the suppressive architecture and then eliminating the cancer cells in the more permissive environment. So, it is more or less like if suppose you have a hard shell and you want to target something that is inside that shell. So, first you have to drill a hole in that shell and then you can get access to the components inside it. A similar kind of approach is being used in the dual targeting strategy. So first the fibroblasts are being melted and then the malignant cells are targeted that way.
Right, right. So now let's talk about CAR T cells exhaustion, which seems particularly problematic in solid tumors, I believe.
Yeah, it
And
is.
how do you think, how do these certain checkpoint pathways specifically limit CAR T efficacy?
Okay. So CAR-T exhaustion in solid tumors is multifactorial, but it follows a predictable molecular cascade. So chronic antigen exposure upregulates PD-1, TIM3, LAG3, and TIGT expression, creating multiple inhibitory pathways, unlike the physiological T cell exhaustion. Now, in CAR-T, exhaustion involves constitutive CAR signaling that mimics chronic activation, leading to transcriptional and epigenetic changes that are difficult to reverse. The microenvironment accelerates this process through sustained inflammatory signaling. So, interferon gamma production by activated CAR T cells upregulates PD-L1 expression in tumor cells, creating negative feedback loops. Also, TGF-beta signaling not only suppresses CAR-T function but also promotes regulatory T cell differentiation from conventional T cells within the tumor. So I think with this we can tackle the CAR-T exhaustion problem that we have in tumor cells, solid tumors specifically.
Right. So I wanted to know that what different engineering approaches are being developed to overcome these kind of exhaustions.
So several innovative strategies are emerging, like dominant negative TGF-beta receptors can be co-expressed with CARs to confer resistance to TGF-beta mediated suppression. Also, PD-1 knockout CARs eliminate a major exhaustion pathway. More sophisticated approaches involve switch CARs that convert inhibitory signals into activating signals, for instance. So CARs that detect PD-L1 and convert that binding into T cell activation rather than inhibition. So this approach is also in process. Such kind of engineering processes are being practiced by scientists across the world to develop and overcome exhaustion of CAR-T.
Right. So I believe antigen escape represents another fundamental challenge. So how does tumor heterogeneity specifically impact CAR T efficacy in solid tumors?
So, since you're talking about tumor heterogeneity specifically impacting CAR T efficacy, solid tumor antigen heterogeneity is far more complex than hematological malignancy. So, the spatial heterogeneity means different tumor regions experience different antigen levels, creating sanctuaries for antigen losses. Whereas temporal heterogeneity involves antigen loss under selective pressure, something that, Darwinian evolution in right time kind of example. But deeper issue is the functional heterogeneity, wherein even cells expressing target antigens may be different, maybe in different activation sites, states or metabolic conditions or cell cycle phases that affect CAR T recognition and the killing efficiency. So this creates subpopulations that survive initial treatment but then can in future regenerate the tumor.
Right. So how are multi-antigen targeting strategies addressing this kind of a challenge that is presenting itself at the very forefront?
So, multi-antigen CARs are essential, but they are technically challenging as well. So, a Boolean logic gates and or not enable sophisticated targeting strategies, or gates allow targeting of multiple antigens, reducing escape probability. And gates require multiple antigens for activation, improving tumor specificity. Gates also prevent activation when normal antigens are not detected, thus reducing on-target, off-target tumor toxicity. Hello.
As we are getting to conclude, what do you see as the most promising approach for achieving CAR T efficacy in solid tumors?
I believe the future lies in the combination strategies that address multiple barriers simultaneously. As we discussed in the other questions, that are dealing with a solid tumor requires multiple faceted approach. So armored cars co-expressing cytokines like interleukin 12 or interleukin 15 can remodel the microenvironment while maintaining CAR T function, whereas combination with checkpoint inhibitors and anti-angiogenic agents or microenvironment modulating drugs create more permissive conditions for CAR T function. But the most exciting development in regional delivery approaches, that is intramural, intrapleural, or intracranial administration that bypasses the systemic trafficking limitations while achieving high local concentration is the thing. These approaches combined with microenvironment preconditioning may finally unlock the CAR T potential in solid tumors.
Dr. Pathak, thank you for this comprehensive analysis. With that,
Okay.
we get to the end of our podcast. And if you like this episode, do not forget to subscribe and like.